Welcome to tonight's lecture by Collin Roesler, our newest William R. Keenan Jr. Professor in Earth and Oceanographic Science. These moments of recognition for our faculty are important ones to celebrate, not only for the honoree, but for our entire campus community, and I'm so glad to see so many people here tonight. They're also a genuine pleasure for me personally, so I'm very glad that we're here to honor and learn from Professor Roesler together. This professorship was established at Bowdoin by William R. Keenan, Jr., Charitable Trust in 1975. The trust's namesake, William R. Keenan, Jr., was a multi-talented educator, researcher, and tinkerer who operated a dairy farm that also served as a research station for scientists. The chair was expressly created to honor and encourage faculty who excel both in teaching and scholarship. And the list of previous Keenan professors includes some of Bowdoin's most accomplished and beloved faculty, including Roger Howell, Jen Scanlon, and the current holder of the other Keenan chair, Thomas Baumgart. The new Keenan professor, the newest one, Collin Roesler, is a truly deserving choice, as I hope you'll see from some of the things that I'm going to share tonight, and also, as I imagine, from the lecture that she will give us. She's the epitome of a teacher-scholar. She conducts research at the vanguard of her field, engages her students in captivating classroom and lab work, and mentors students who are producing important work themselves, and who are often inspired to go on to their own careers in science. Collin studies ocean ecosystems, specifically using environmental optics to examine the responses of ecosystems to environmental pressures. Her research extends from the depths of the ocean to outer space. To give you a sense of the way she engages with this work, in 2018, she lived for 37 days on a 238-foot research vessel in a remote patch of the Gulf of Alaska, some 200 miles from shore, studying plankton and bacteria. This is just one of the many such trips she has made over her career. Collin is currently part of the research team for one NASA's space satellite, which was launched into orbit just last month and will use imaging equipment to scan the Earth to monitor changes in ocean plankton and other marine plant life. In addition to her work with NASA, she's received research funding from the National Science Foundation, National Oceanic and Atmospheric Administration, the EPA, the Naval Research Laboratory, among others. She also published her research widely in top journals in her field. Collin has long been known as a committed and compelling teacher as well, something that her students recognize and appreciate. In 2013, the senior class selected to give her the annual Karofsky Faculty Encore Lecture, based on her excellence in teaching and influence on students as a mentor and role model. Here's one example of Collin's excellence in this space. In 2017, Collin took 3 of her students to Hawaii, where they presented their honors and independent study research as the ASLO Ocean Sciences meeting in Honolulu, a rare opportunity for undergraduates. A few years later, Collin co-published significant research with one of those students, Luke Carberry, class of 2018. The paper, which designed a new, more accurate method for calculating the amount of phytoplankton in the ocean, was published in the journal Limnology and Oceanography Methods. And I overheard a number of students who came into this hall this evening talking about them being a fan club from a previous iteration of one of Collin's classes. Collin earned a bachelor's degree from Brown University, a master's degree from Oregon State University, and a PhD from the University of Washington. She's a groundbreaking researcher, a devoted teacher, and an inspiring mentor, and I am very glad that we are honoring her tonight. Please join me in congratulating and welcoming to the stage the William R. Keenan, Jr., Professor in Earth and Oceanographic Science, Collin Roesler.
Thank you, Safa. I think it's really unfair that then I have to talk after that kind of an introduction. I'm really honored to be here tonight, and I'm really honored to have this professorship and that I share it with Jen Scanlon and Thomas Baumgart. I wanted to just comment on this. I'm going to move out of the light, sorry. That's kind of ironic. I wanted to just comment on this photograph. This is taken from the North Atlantic in 2021. We were on a research cruise in the North Atlantic on the British research vessel Discovery. I had the night shift, so I have a lot of nighttime photographs. This is just one of them. Okay, so I'm going to give you just a little outline to my talk tonight, and I'm going to do it in a series of little vignettes, and I'll comment on what a few decades and now endless possibilities mean. Oh, that is not good. Those are supposed to be down. So the first one is talking about how I discovered climate drivers and ecosystems and how these climate drivers change ecosystems. And I am thinking about this from my own experience as a student, so the classes that I took, and then how that has informed the classes that I teach. Then I'm going to jump right into some algorithms, which is a fancy way of saying the way that I solve problems. And then validation, which is the way that I test the way that I solve problems. And then that will lead us into endless possibilities. Okay? So John McPhee is one of my favorite writers. He's a nonfiction writer who talks a lot about science. But this is actually my favorite book of his. It's on the writing process. He's a professor of writing at Princeton, or was. He's emeritus now. But my favorite chapter of this book is called Structure. And so he talks about how you weave a story together when you have a clear chronology from past to present to future. And he says, so we're dealing with this journey in a certain piece of time, and could we possibly bend it? And by bend it, he means wrap it, right? And if you wrap it, then you can decide where you want to come into this story and where you want to go with this story. So I'm using that so that I can start in the present to kind of get us all up to speed, go to the past, and then it'll bring us into the future, okay? Get that book, it's like really, really amazing. Okay, so at 1:30 in the morning in February, NASA launched the PACE satellite. And it was aboard the SpaceX Falcon 9 rocket. And I don't know if you can lower the lights a little bit just so we can just get a sense of how amazing that rocket is as it's taking off. Thank you. And then this is what it looks like when it broke through the clouds. And this is actually going to be the satellite that makes it up into orbit. So PACE, which is a bad acronym, and I didn't make it up, the Plankton Aerosol Cloud Ocean Ecosystem. The most important part doesn't get a letter. It's kind of, again. It has sensors that collect data that are going to be used to describe not only the concentration of phytoplankton in the ocean, but also their composition. And it's going to do that on the global scale, and it's going to do it with 1 kilometer resolution, and it's going to do it every day. So that's amazing. Right? Let's break that apart, just to make sure. Phytoplankton are single-celled, microscopic photosynthetic organisms. They are the base of the marine food web. Microscopic. Satellite. This space-time scale that it will resolve, remembering that these satellites orbit the Earth every 90 minutes. It's really fast. And at an altitude of 700 kilometers above the Earth. Right, so again, this is an amazing feat. The sensors that collect this data, because the satellite is above the atmosphere, are looking through the atmosphere, but the atmosphere has a lot of signal. It sends off light that gets detected by the satellite, and in fact, the signal is 90% atmosphere and maybe 10% ocean. And so we have to get rid of the atmosphere. I'm not going to talk about it, but if you want to learn about it, take my remote sensing class in the fall. You get to really delve in. So this is an amazing feat. Now, it turns out I have been waiting for this satellite my whole life. And I was fortunate enough to see the launch. So if you ever get an opportunity to see a launch, even if you're not involved in the satellite, you should do it. It's awe-inspiring. Okay. How does this work? Well, I'm going to show you a little animation. This is data that is from satellite. There is data for land, which is fine. And then there's data from the ocean, which is great, and also ice. It's going to show you an annual cycle of the biosphere. And so what you'll see is how spring arrives in the ocean and on land and moves to the north, and then how the winter comes to the northern hemisphere and spring arrives to the southern hemisphere. So the colors are intuitive. So there goes the spring into the Arctic and the ice in the Antarctica. And now winter is going to come to the north and spring to the south, right? I know, it's beautiful, right? Okay, so amazing people, visualizer, scientist, and producer of that image. Thank you for that. Okay, so how does this work? Well, the color of the ocean varies. And if you go out to sea, most of the oceans and the subtropical latitudes are pretty blue, right? And they're not blue because it reflects off the sky. I'll show you why they're blue in a minute. But as you head into productive areas, say in the subpolar regions, or you head to the coast, the waters get greener. This is off of South Africa in the Benguela. And it's sometimes hard to see because there's a lot of reflection of skylight off the surface, but if you're in a ship and a wave washes over, you can see the green color, even though it's kind of foamy in the waves. A couple of days later, it looked like this. So there's some big changes in the color of the ocean. And most of this is attributed to phytoplankton, those single-celled photosynthesizers. And we use their dominant pigment, chlorophyll, as a proxy for concentration because all phytoplankton have chlorophyll, and really only phytoplankton have chlorophyll. So it makes it an easy proxy. And probably everyone has heard chlorophyll. All right, so now I'm going to give you a short primer on ocean color. And ocean color is actually a technical term. It's a satellite term. It's different than the color of the ocean, which is kind of a poetic term. So we're going to talk about ocean color here. So what I'm looking at here is essentially a cross-section that I've created through this image. So the sun is white. We're all aware of that, even though we're not supposed to look at the sun until next Monday when there's an eclipse. And if you're in the zone of totality, you'll be able to actually look at the corona for a minute. Sun is white. That means that the colors coming out of the sun are basically evenly distributed across the spectrum, mostly. When it goes into the ocean, water absorbs mostly red light, and it looks something like this. So the absorption means that light of that color doesn't penetrate very deep, but that colors that aren't absorbed penetrate very deep. In fact, maybe down to 200 meters in the ocean. Particles in the ocean backscatter that light out the surface, a fraction of it. And so you can see the shape of the light coming out compared to the equal distribution of light going in. Well, and that's where a satellite says, great, and it's measuring that light coming out of the ocean. Okay? What it measures is the spectrum of intensity, and we do some normalization, but essentially, we look at a spectrum that looks like this, and if you don't read or speak in nanometers or wavelength, I'll just put a spectrum down there. But what you can see is most of the light coming out of the ocean is in the blue, and that's because water absorbed the red light, okay? And that's why the ocean looks blue. So this is a really intuitive field, right, when you study ocean color. So that's what most of the sort of subtropical waters look like. When we head into the coastal waters, we still have that same white sun, but now phytoplankton absorb blue light. And you can see that it just doesn't penetrate very deep. And so what's penetrating deepest into the ocean is the green light. And then particles scatter that back out, and satellites measure it. And the spectrum looks something like this. And so I've left both spectra on there so you can see that not only is it getting darker, but it's getting greener. Right? So now we go to the red water, right? And in the red water, there's a lot of phytoplankton. This was a harmful algal bloom off of Africa. And so there's a lot of absorption. In fact, there's so much absorption that we see only like the sort of yellow to orange to red colors making it, and they don't, again, make it very deep. Particles will scatter it, the satellite will measure it, we get the picture here, and this is what the spectrum looks like. It's very dark and it's very red. And for those of you who are interested in physiology, that's actually chlorophyll fluorescence. The fluorescence by chlorophyll is so strong, we actually can measure it coming out the surface. So we make use of this sort of shifting in intensity and shifting in wavelength to tell us the concentration of phytoplankton in the ocean. And a colleague of mine, Kurt Mobley, did a really nice model where he looked at a continuum of these spectra as a function of chlorophyll concentration. So when you have really low chlorophyll, it's blue and bright, and when you have a lot of chlorophyll, it's dark and red. And so in 1978, based upon this idea that waters get greener and even into red, the more phytoplankton you have in the water, if you could measure the greenness from satellite, you could estimate the concentration of phytoplankton in the ocean. And so that satellite was launched in 1978. It had 4 channels. And the current way that we talk in NASA about channels is by talking about crayons. So think of a crayon as representing a single color and a single point along that axis, and that's all you get to see. So your data suddenly now look like this, right? It's really nice when you have the full spectrum, but we don't have the full spectrum. Satellites don't have the full spectrum. Oh, and yeah, we got to use that red channel to correct for some of the atmosphere. So really we have 3 channels to determine greenness. So imagine you have an image, and this is like a swath from a satellite orbit. It's North America. In Central America, it's a cloudy day. This isn't the best image. It's a true color image. But imagine that you could look at the light coming out of that surface with glasses that have a filter on them that kind of keep out everything but some small wavelength band. It would look like this. So it's the same image with blue glasses on, with teal glasses on, green and red, okay? And if you could look up close, you would see parts of the land are brighter, parts of the ocean are brighter. But it's being able to determine at each location on the planet what that spectrum is. You can estimate the chlorophyll concentration at that location. And so that's what we do. So all the images you see of chlorophyll, or they sometimes call it primary productivity, which it isn't really, but all these images that show you the productivity of the ocean are really just measuring the greenness of the ocean. So that's if you have 4 crayons, 3. What could you do if you had more? I'm not going to answer that yet. Okay, so now I'm going into the vignettes. That was the present. This is where we are, okay? So now I'm going to start and think about, sort of on decadal scales, how I discovered climate drivers and how they impact ocean ecosystems, and I did this in classes. And how my experience in my own classes informed how I teach my classes here at Bowdoin. Okay, so I'm going to give you a couple of examples. Then I'll talk about, again, the algorithms, validation, and possibilities. So when I was a senior in college, I took a class in weather and climate from Tom Webb. And he gave us an assignment, and I'm looking at my students. We had to do a daily weather journal. And what that meant at that time was we had to go to the newspaper and cut out the map of barometric pressure. And I actually found one from just about that date. I graduated in May of 1985. I guess there was a big storm. I missed it. And so I cut them out, and I pasted them into a notebook, and I color-coded high and low pressure because I was trying to make sense of it. And I actually made a flip book so that I could like flip through. I know, right? I could flip through and see how the weather changed over the 3 months of the class, right? It was an amazing experience for me because it gave me a chance to connect what an atmospheric pressure cell was to what my local weather was. And when it was a high pressure system, it was a sunny day. And when it was a low pressure system, you better bring your raincoat. And when those isobars were packed together, you should fly a kite. And that intuitiveness that this sort of mundane, everyday, cutting out the weather map really struck me in terms of thinking about how to make observations. It also, the flip book, allowed me to watch how these systems moved across the country and how everything always came from California, because we are in the latitude band of the westerly winds. So systems move across, right? And I got to get a sense of how fast they moved across, right? Winds are very fast, but systems move slowly. And so again, I got a really nice intuition about that. And so it came to me that although science can sometimes take a long time to accumulate observations, you sometimes need a lot of observations to be able to find meaningful patterns. And so that was a lesson that really struck with me. So I teach introductory oceanography, and I have a few students in the audience. And they recognize that I have transformed this into a daily moon journal. My students have to go out and look for the moon every single day. And it can be really frustrating when we start this at the end of January because it's cloudy. And they have to identify the phase of the moon. And they have to write down when they see it and what angle it is in the sky. And that way you can calculate what time it's going to be directly overhead, which is called the apex hour. And so once they finish doing this for 6 weeks, we come back to the lab and they start to do an analysis of local tides. And what they find, they've already turned their assignment in, so it's okay for me to give the answer. What they find is that when you look at the time series of the range of tide heights, it is totally related to the phase of the moon. Full moons and new moons, you get really big tidal range. And it turns out the hour that the moon is overhead is about when you get a high tide, and then 12 hours and 24 minutes later. And so really all you need to know is the phase of the moon, and you can predict everything there is to know about the tides. And so doing this for 6 weeks gives you an intuition of where's the moon and when can I find it. The other cool thing that we do with this assignment is that we link it to the synchronous coral spawning of the Great Barrier Reef. And corals spawn basically one day of the year at one time of that day in one phase of the tide. And the analysis that you do in Harpswell can be translated exactly to the Great Barrier Reef, and you can predict the timing of this spawning event in case you ever want to go and dive the Great Barrier Reef and see it. You just need to know the moon phase. You can figure this out. So that allows me to kind of think about how physical processes drive biological ones, kind of get students going on that. The other assignment I had to do for this class was a final paper, a literature-based paper. And I got really excited about El Nino. And it's not like we were I'm not that old, but we knew a lot about El Nino, but we didn't know everything about El Nino. And it's fascinating. It's where the trade winds in the Pacific weaken, sometimes even reverse. Those winds have pushed water to the western Pacific, and when they die, that water sloshes back. And slosh is a technical term. You find it in physical oceanography journals. That water sloshes back against South America and then spreads up and down the coast. So I was looking for a paper. We used to have to go to the library to do that. And the most recent edition of the paper of the journal Science had this on the cover. And it said, satellite color observations of phytoplankton distribution in the eastern equatorial Pacific during the 1982-83 El Nino. And the abstract goes on to say that the change in the concentration and distribution of phytoplankton had really serious trophic impacts for animals and even up to seabirds. And so again, here was this situation of climate scale phenomenon driving ecosystem response that I got really excited about. And then I had to learn something about ocean color and how do you get phytoplankton from a satellite. And what the satellite showed was in the Galapagos Islands, here are the islands, under normal situations, the red is high concentrations. There's a lot of nutrients on the western side of these islands, but when these winds reverse and this warm water sloshes back, you see within weeks a decrease in the concentration And that's why you get this sort of trophic cascade. So I wanted to delve right in. You can write a literature paper, but you can also do data analysis. And my professor was like, well, then just figure out who to ask for some data. And so I wrote a letter, because there was no email, which is crazy, I know. I wrote an actual letter to Dr. James O'Brien at Florida State, because I figured out that he had wind data. And he sent me 20 years of data from 126 stations in the equatorial Pacific on a tape for a PDP-11 computer. So that was crazy. And then I asked another professor at Oregon State for sea level data from California to see if when that water sloshed, whether you could see the signal in California. So, turns out wind data is really noisy and really hard to work with. So I had to learn some statistics, but I did. And then I got practice writing a scientific paper, and I found it a couple of weeks ago in my office with the grades from Professor Webb. My discussion needed some work. But it was amazing because it taught me that you can jump in with data and really investigate how biological systems respond to this forcing, but that you really need good data sets. And you need sometimes big spatial data sets like you would get with satellite, or you need long time series that you might get from moorings. And so that kind of stuck in the back of my mind. I went into grad school and I had to take my core courses. One of them was marine zooplankton, and we had to write a paper. And I wanted to do some data analysis. So I asked my advisor, who was not a biologist, do you have any data on zooplankton that I could look at? And he introduced me to CalCoffee, which is the California Cooperative Oceanic Fishery Investigation, which was brought together to try to study the collapse of the sardine fishery off of California and Baja. It's an amazing program. This is their sampling strategy for ships. Those are stations, and they visited them monthly for a while. That's an incredible, incredible data set. It got to be quarterly after a while. 32 years, and they measured zooplankton, so I was in heaven. So I discovered two things while I was doing this class project, and basically it ended up being my master's degree because, because it did. So the first thing is that in the normal situation off of California, the California current flows down California, and the water's cold and it's rich with nutrients because of upwelling. It's very productive. The zooplankton concentrations are very high. But the species are northern species, cold water species. And then down on the southern end of Baja, the water's warmer, there's a lot less nutrients, it's not as productive, southern species. But during El Nino, with that sloshing warm water, that water pushes its way up the coast. And so this low concentration and southern species push that boundary up to California. And it turns out that it's related to El Nino, as we expected, but we could actually demonstrate this with this data analysis. So these boundaries move back and forth, and when El Nino comes through, sea levels are higher, that water is warmer, it expands, it sits higher. And so when we see a positive sea level anomaly, and I shaded the El Nino events, we also see very low zooplankton. And so that was a pretty exciting result. And it turns out that this region here that was shaded underneath represents that area for which this pattern of variability shows up the most. And so what it says is that this is the hot spot, and we interpreted this as that boundary moving back and forth, right? Okay. So then the second thing that I discovered in this analysis by mistake, because I didn't log transform my data when my advisor told me to log transform the data, because that's what you do with biological data, and I forgot. But I discovered something cool by doing that, because sometimes science can be by accident, but it doesn't mean it's not useful. So there's some features along the California coast called coastal squirts and jets. Again, technical terms. Associated with wind-driven upwelling, where you get nutrient-rich water brought to the surface, and they just shoot offshore in these thin plumes. And thank goodness for remote sensing, you can see these features in phytoplankton. So there's Point Conception, there's San Francisco, that's San Diego, and you can see these big plumes coming offshore that have really high phytoplankton concentrations. And again, going to the library, oh my gosh, I sound like Hermione. Going to the library, in the restricted section, I found this paper that shows this chlorophyll concentration that explained this interesting feature in zooplankton that we only observed when I have these arrows, these times of really high biomass. And so it turns out when you have these big events, the phytoplankton are transported offshore and the zooplankton take off. And you get these offshore ecosystems that are totally isolated from the coast. So that was fun. So thinking about how much I loved that kind of work, I decided to bring this to my classes. It was an inspiration for my course, Ocean and Climate. And the learning goals of this course are focused on data collection and data access and data curation. And the skills are focused on computational analysis and how you take data sets and isolate different signals, in particular climate signals. So 2 examples from this class are, 1, I give students 20 years of hourly observations of physical properties and biological properties that are measured from 8 moorings, the ones that are red in this map. And my colleague and I, Neil Pettigrew from University of Maine, deployed these moorings in 2001, and we've been collecting data ever since. It's a really astonishing data set. And so students can use these data to look at seasonal cycles and how those seasonal cycles have changed over the last 20 years. They can look at decadal trends and think about what might be happening as the Gulf of Maine warms, which it is. The other data set I give them is a 6 million year record of climate from a deep sea sediment core that my undergraduate advisor sent to me when I got the job at Bowdoin. And he's like, yes, take this data set. You can do so many amazing things. And what we can do is students can learn some different techniques to pull out the cycles of ice ages that are driven by orbital changes, and it creates these cycles of glaciation, and we can, in fact, even pull out signals associated with tectonic-driven climate change. Okay? Excellent. Okay, last class. My advisor, after I finished the zooplankton paper, was really wanting me to get on ocean color. I was so excited to start. But we didn't know everything about ocean color. He was really an altimetry guy. So he sent me to a summer course up in Friday Harbor, University of Washington, to study optical oceanography. Because it turns out phytoplankton aren't the only thing that turn water green. And so if there's other things in the ocean that absorb blue light, the water turns green, and we call it phytoplankton, and that's not true. So he sent me off to figure this out. That course really changed my life. It completely redirected my graduate program. I finished my master's thesis with the zooplankton paper, and I got my butt up to Washington to join Mary Jane Perry's lab to study what are the biological properties we could pull out of satellite data. It also changed my summers, because after taking that class, I TA'd that class, and then I taught that class. And so over the last 30 years, it's taught every other year. I basically spent 15 summers out of the last 30 years involved in this class, which started off in Friday Harbor, moved to the Darling Marine Center, and then most recently, the last 2 times we've taught it have been at Schiller. Where's Holly? Here she goes, yeah, Schiller. So it was a great experience, and it is the basis and inspiration for my remote sensing course, being taught in the fall, if anyone wants to take it. Okay, so that is the deep dive into classes I took, how it informs my teaching, and really thinking about, scientifically, the interaction between physical drivers and biological response. So then the next decade was really about algorithm development. And so I was thinking about if the ocean has a certain color and we can measure its spectrum, we should be able to figure out who's in the water. Because we know that phytoplankton absorb, and we know that particles scatter. And so figuring out a way to take those properties and predict the spectrum associated with that water. We have something called the radiative transfer equation. It's a really complicated equation that describes exactly how water moves through the ocean, gets absorbed and scattered in all these different directions, but we can't solve it. It's got a derivative and it's got an integral, and you can't solve it analytically. So we make some approximations depending upon our situation and do the best we can. And so for looking at ocean color, we can make an approximation that tells us that if we actually look at the ratio of backscattering overabsorption, and those are summative, so we can just add up all the components that scatter and add up all the components that absorb, we should be able to model the spectrum of light coming out of the ocean and being detected by satellite. So this is my pictorial equation. There's your division line, just like this. So we know that water is a big absorber, and so that's one of our components that's going to be in the denominator. We know that phytoplankton absorb light. That's going to be another component in the denominator. Other particles also absorb light. That's one of those components. That's a problem. And if you're anywhere near the coast where there are rivers like Maine that are really brown with tannic acid, that absorbs light, okay? Water molecules, when they cluster together, scatter light, so they're in the numerator, and then any other particle might scatter light. But these are all additive. And so if you could identify how much of each one of those, you can mix them together in different ratios and reconstruct different color spectra. Okay? That was the idea. So I spent a long time figuring out what those properties were. So here's the graphical equation. These are the spectra of absorption and scattering, and I spent a long time figuring out how to make those measurements because seawater is complicated to separate them all out. And then I developed a model where essentially you allow these all to vary and try to reconstruct whatever your measured spectrum is. And then I went out and I measured a bunch of spectra from different environments. They're really different in magnitude and color. I modeled them. I picked these 4 different ones to show you. The colored line is the measurement. The black line is the model. It doesn't do a good job where there's fluorescence, but it does a pretty good job in other places, which says that those few components explain most of the color variations in the ocean. They just come in different ratios. But then you have to validate, so like how much phytoplankton absorption did you estimate versus how much you measured, and it's not bad. I mean, it's a large dynamic range, lots of orders of magnitude. And same thing for scattering. But what it means is that that model works, that really simple approach can do a lot of explanation. And you can separate phytoplankton from the other greenness in the ocean. So time goes by, they put up more satellites, they add, a couple more crayons. Not enough. But I was thinking a lot about phytoplankton, and this is from an experiment that I did with Chris Proctor, who was a former Bowdoin student when he was my master's student, and he took this picture of our cultures. These are different species of phytoplankton, and you can see when they grow to really high concentrations, they have dramatically different colors, right? That's because they have dramatically different pigments, which means they have dramatically different absorption spectra. And so if you could separate phytoplankton based on their absorption spectra, then you should be able to see that in the different hues of the ocean. It's green, but is it blue-green or is it yellow-green, right? And so you should be able to get phytoplankton community composition if you had enough wavelengths, right? All right, so we went to test this. We were invited by some colleagues to go to South Africa, to the Benguela upwelling system, which has recurrent red tides. And when I say red tides, I mean red tides and yellow tides and green tides, and the species composition changes, and it is dramatic, and they're very high concentrations. And so my colleagues were Grant Pitcher and Stuart Bernard. This is a beautiful image of these red tides in this region. And so thinking about, if we knew these absorption spectra, could we use that to model this color? And so we did. So we went out there, and the beginning of the time we were out there, the water was green. By the time we ended our experiment, it was red. We measured the spectrum. And in the beginning, and we separated it into how many of these 4 different groups of phytoplankton might be mixed in to these blooms. And in the beginning, there wasn't much of anything out there. And that's the spectrum. And our model tells us there wasn't much of anything. Then the water turned this maroon color. And I don't know, Heidi, if you remember, it was like the color of red wine. And that spectrum is crazy. And that spectrum is telling you there are a lot of pigments. Then the water evolved into more of a greenish color. And again, we saw different predicted concentrations of these 4 phytoplankton groups. And then at the end, the water was like rust. And so we have this result. There's this changing ecosystem based upon this model. But how do we know we're right? Well, luckily, our colleague Grant Pitcher collected water samples while we were out there, and he looked under a microscope. And he validated what the results were. They're not perfect, but what you see is that these are very different scales. And when we see peaks in estimated concentration of a group, we also see it in the microscope. And so that reminds us how important it is to validate, and that you need to develop an infrastructure to validate your models and to make sure that you're right. So why do we care? Why do we care? Why are we trying to do this work? Well, this was a particular situation off of Africa, but globally, diatoms, which are phytoplankton that have glass cell walls and are incredibly beautiful, they are responsible for the majority of carbon pulled out of the atmosphere and sunk to the bottom of the ocean. They are responsible for taking care of a lot of what we do to the atmosphere. They're trying to help us take it out, right? So knowing the distribution of phytoplankton or of diatoms is very important. In a sequence through a spring cycle, diatoms tend to die out when silica is used up because they can't make cell walls anymore, and dinoflagellates come in. So that transition between those 2 groups tells you a lot about the nutrient status and biogeochemical cycling. People care about that. Mesodynium is a photosynthetic phytoplankton that then suddenly starts eating phytoplankton. And it does it when the ecosystem is in imbalance. So when you see mesodynium, it's telling you that something is funky with the marine ecosystem. And then finally, dinophysis. Dinophysis is a toxic dinoflagellate that causes it causes diuretic shellfish poisoning. So you like to know when that's in the water, right? So that's why we care who's in the water. Okay, validation. So we start with phytoplankton, and this is a sort of a forward approach. Phytoplankton are in the water. And this image is from an automated microscope that we have at Schiller Coastal Study Center. It's from an instrument that a colleague of mine invented. So it measures an image of every single cell in the water, and then it has a classifier, and it tells you what species they are. It's like crazy amazing. So we know who's in the water. We know which groups have which pigments, and therefore what color they are. That allows us to say something about what the absorption spectrum should look like. And then that will then determine the hue of the ocean, and then a satellite measures it. But what we want to do is go from here to here. So we have what's called an inverse approach. We have satellites measuring ocean color. We estimate from that the contribution of absorption to that color, and we estimate the phytoplankton part of all the other constituents part. And that allows us to get to pigments. And then pigments, because they're taxonomically distinct, that allows us to get to phytoplankton groups. Now, I bolded these terms because there's a lot of steps in this modeling approach, and every step needs to be validated. So we need to validate what we measure down here with this little buoy relative to what a satellite sees. We need to validate the absorption that we are retrieving. We need to validate the pigments that we think we're retrieving. And we need to make sure that we have those cells in the water, right? So that's what I've been doing in the last part of my career, is figuring out ways to validate. So was part of the PACE science team, and my job was to develop protocols for validating these products. So we measure ocean color aboard a ship, and there's a picture of Sue and me in the North Atlantic on a sunny day, measuring the light coming out of the ocean. And we developed an underway bio-optical system based upon my colleague Emmanuel Boss's design. And water comes in through the bottom of the ship, flows into the lab, flows through all of these sensors, gets partitioned through filters so we get different sizes. We measure the absorption properties, the scattering properties, the fluorescence properties. It goes into an IFCB. We measure the cells. Most of these instruments we also can do from a mooring. So this is the Bowden buoy. For those of you who have not visited the Bowden buoy, it sits out in Harpswell Sound and it measures bio-optical properties every hour. And then we also have to validate with pigments. And so I don't run an HPLC, but Emily Collin runs the HPLC. And she teaches all my students how to run the HPLC so that we can validate this estimation of pigments. And then we run through the instrument through the imaging flow cytobot, which is a robot, and that tells us who's in the water. Okay. So, endless possibilities. PACE is up. PACE is in the sky. It detects ocean color. Doesn't everybody want one of these, right? Like, everybody's like, oh, I could do some coloring. It is measuring the color of light coming out of the ocean at over 200 different wavelengths, which is about 5 times that many more colors, right? So when I show you this image of chlorophyll concentration, what PACE will be able to do is provide you with a global image where instead of just chlorophyll or concentration of phytoplankton, it will tell you the concentration of phytoplankton groups taxonomically. Diatoms, dinoflagellates, dinophysis, mesodynium, and all the others that we can detect because of their different pigments. Right, I know. And so as we have PACE scan the globe and return data on the spectral light, this is showing the animation of the seasonal biosphere. But imagine that you could get a seasonal cycle of these unique phytoplankton communities and see how they change from winter to summer. But now we would let PACE stay up for a little while, and we can begin to see how these populations change under a changing climate. We don't see a lot of phytoplankton in the subtropics because they're very nutrient limited there, but we see a lot of phytoplankton up in the subpolar regions. But as the climate warms, those subtropical regions expand. And the subpolar regions are either going to expand to the Arctic or they're going to compress. And those are our big productivity zones. Those are where the major fishing zones are. Those are where the major carbon drawdown zones are. And once we have the capability of being able to detect these different groups and look at how those communities expand and contract with climate change, I mean, we will have knowledge beyond what we can imagine. And so the light has arrived, right? And science is no longer limited by technology. The decades that I waited for more wavelengths, it's over. That wait is over. And now PACE, for me and my colleagues and for you, represents some limitless ideas. So it's a pretty exciting time. Okay, I have a few people to thank. And my dear friend Jen told me, practice this, because this is the roughest part of the talk. I have had an amazing set of advisors. From my undergraduate advisor, Warren Prell, who was a paleoclimate reconstructor, to Dudley Chelton, who was my master's advisor, who was a physical oceanographer, tough guy, but man, I learned a lot from him. To Mary Jane Perry, who was my PhD advisor, who taught me everything from phytoplankton physiology to remote sensing. And to Ron Zenefeld, who was my postdoc advisor at Oregon State, who is a brilliant physicist and a brilliant painter. In 2018, Mary Jane and I ended up on the same research cruise. It was a NASA-funded expedition to the North Pacific, and we got to share a cabin for a month. And it was really great. We had so much fun. She continues to be an amazing person. I've had not that many graduate students because I've been here and we don't have a graduate program. But when I was at University of Connecticut, I had 3 graduate students who were amazing. Jeremy, was my first master's student and my last PhD student. He was a PhD at University of Maine, and he took a lot of time off in between. He is now the project scientist for PACE at NASA, so I really couldn't be more proud. And then I would just like to also highlight Chris Proctor, who was a Spoden graduate of 2005, who's also working at NASA now. And then Andrew Barnard was my postdoc, who was on board when we were developing the moored array for the Gulf of Maine. And he literally developed all of the hardware and firmware for data and power for these moorings in his barn in Alna, where we live, which is 40 minutes away. And we still use this 20 years later. It's so amazing. And then I have undergraduates. So these are all of my undergraduate honors students, or students who did advanced independent studies with me, starting from the early days, Dan McGrath, to the later days with Lyle. Each one of these students worked on a validation project, or an algorithm project, or a ecosystem project. And look how happy they are. They're amazing. Each one of them has done like amazing work. And my department. I joined the department when it was the Department of Geology, and they welcomed me, even though I was an oceanographer. And it has been an amazing place to work. It's an amazing group of people who support each other in our scholarship and in our teaching and in our life. And so I couldn't be more grateful for this department. I have had 3 technicians. I've had people kind of come in and do little bits here and there, but I have had 3 technicians over my entire career. And boy, stuff wouldn't get done without them. So Jennifer Simeon joined me when I was at University of Connecticut. She was right out of undergrad. She took my biological oceanography class and signed up. So she got me going. She helped me get my research going. We started doing field programs together. And that was a wonderful experience. And then I moved to Maine. She moved to graduate school. And that's when I hired Heidi Franklin, who's here today. This is a picture of Heidi in the Benguela Current off of South Africa, happily smiling at the dinophysis that she's captured. Heidi was there for the development of the moored array and for figuring out like how we were going to keep this program implemented forever, and it has been forever. She went on copious field trips with me, and when we got too busy, she hired Sue. She came all the way through Bigelow Lab. We spent about a year at University of Maine before coming to Bowdoin College, and she had to head off to Australia. But she left me with Sue, and Sue has been with me ever since. And she took over all of the responsibilities. And being at Bowdoin, teaching takes a lot of my responsibility, which means a lot of it falls on Sue. And I will just say that science gets done because of Sue, and I am so grateful. So. It's hard to be a field oceanographer because you're gone a lot. I think my first year in Maine, I was gone 7 months. My husband's saying, yes, she was. But he gets it. He's a field scientist as well. And so we swapped Who's responsible for what? And my kids just moved right into that. When one of us is out of town, someone's cooking, someone's feeding the pigs, someone's collecting the eggs, and they just roll with it. And I'm so grateful for their support, because if you don't have that kind of support, it makes it really hard to be productive in the field. And then I'd like to thank my mom. My mom raised me by herself, and she told me, go for it. Never let anything hold you back. She was an English major and an artist, and she did not know where all the science was coming from, but she supported it. And I know that she would be really excited to know that we both love light and color. So, thanks. So, if you have questions, we have microphones, and it always takes people a little bit of time to try to get their thoughts. So I'm going to tell you a little bit about this picture. This was from the North Pacific part of the NASA program. So we spent a month in the North Atlantic and a month in the North Pacific. And in the North Pacific, it was cloudy every day for 32 days. We called it 32 shades of gray, but not as fun. On the day that we left, and we also didn't see anything. We saw no whales, no dolphins. A piece of kelp floated by, and 50 scientists flocked to the side of the boat, and we fished it out, and we looked at the organisms that were growing on the kelp for hours, right? On the day that we left, the clouds parted, and a pod of hundreds of humpback whales came to the boat and frolicked around us and fed, and as we sailed home to Seattle. So anyway. Did I answer all your questions? Yeah, go for it.
Hi, Collin. Thank you. That was very eye-opening. There were a lot of aha moments.
Thank you.
I was just wondering, what are some of the other things that make the ocean green besides phytoplankton?
Yeah. So the organic matter that comes out of rivers, we call it colored dissolved organic matter because we're clever. It also, anything organic absorbs in the blue. And so when that comes into the ocean, it makes the ocean green. And so the Gulf of Maine is very, very green. And some of it is phytoplankton, but a lot of it is the materials coming out of the rivers. And so if you look at images of chlorophyll, they'll be really high chlorophyll on the coast. And I'm really high CDOM at the coast. It's not chlorophyll. So that's one thing. And the other thing are just other particles. Zooplankton, bacteria, detritus, it has organic molecules in it too. And so you just need to separate those from the phytoplankton. And so that's sort of that tricky thing. But so yeah, if you look at the Gulf of Maine, if the water is clear and green, it's CDOM. If it's murky and green, it's phytoplankton.
Cool.
Yeah.
Thank you.
Mm-hmm.
Thank you so much, Collin. That was fascinating. I was especially interested in your finale, which was the big picture of why all this matters. And I was curious if you could say a little bit more about this piece of the climate change puzzle, because it's so interesting how you're contributing to our understanding of what's happening with climate change. But I'd also be curious to hear you talk a little bit about how the research that you and your team are doing interacts with other researchers' work.
So people are really interested in how these ecosystems are going to change as the climate warms. And so one of the things about those subtropical zones is that it's very warm there, it's very stratified, and there's not a lot of mixing, which means there's not a lot of nutrients being brought up, right? And as we warm, that characteristic is propagating. It's propagating to the mid-latitudes, which means that the organisms that do well in that environment are going to propagate also. So that's sort of one aspect. And the fact that you could expand this subtropical zone means that you are decreasing the productivity, you're decreasing the carbon export, and you're changing the animals that will fall along from the changes in phytoplankton. So that's kind of the one question that people are really interested in. They're also really interested in what's happening around the Arctic, because one of the other things we're doing is melting sea ice. And they're discovering like the expansion of species of phytoplankton into the Arctic that were never there before, because we're changing the biogeochemical cycling, we're changing the nutrient, the stratification, and so those species are starting to move into the Arctic, which means that the things that eat them are moving into the Arctic. And so we're changing an ecosystem in a way that we don't know all of the implications of that. And to say nothing of the fact that when you move sea ice, you can start bringing mammals in, that marine mammals that can need to breathe. And so they're starting to move in too. So we're really changing the ecosystem from the individual, like single cell photosynthetic organisms all the way up to the large mammals and sort of apex predators. And so there's the climate change, like how is the ocean responding to a changing climate, but then there's this, how are the ecosystems responding? And so that's really what my colleagues and I are really interested in, is the large scale, but then also in specific areas, you know, along the coasts of these regions where people make their living and feed themselves with the ocean. Like, what does that mean for communities? And so hopefully we'll be able to answer some questions or at least provide some predictions. So, thank you.
That was an amazing presentation, Collin.
Thank you.
And you're a really good speaker. Holy cow. It's true.
Thanks.
Yeah. So yeah, I'm a political scientist.
I know.
And so it's really exciting for me to speak with a real scientist. My question is, we've been talking about using light to measure,
Phytoplankton.
there, okay. How would the analysis be different if you were looking at fresh water instead of ocean water?
Okay, so here's what's really cool. Well, not that it all isn't cool, but when you get into lake systems, it often has a lot of sedum. And so the lakes can be very dark and brownish in color because of just dissolved organic matter. They're also small. And they're surrounded by land, which is really bright. So when a satellite looks at a lake, it sees a lot of interference from the surrounding land. So it's getting a lot more light, and so you have to do some masking. So there's another satellite that we're hoping to launch, which is in a different orbit, one that allows it to stay above the Earth as the Earth rotates. So it basically orbits around the middle as opposed to around the top. The orbit around the top, the Earth spins underneath it. The orbit around the middle, if you can orbit at the same rate that the Earth is spinning, you can sit over a single spot. And so you can measure multiple times per day, and you can also get because you're getting so much signal, you can look at very small spatial scales, like meters. And so now you can begin to look at lakes, right? And you can begin to sort of look at all of these biogeochemical cycling and all of these ecosystems on the scale of lakes, on the scale of 1 to 10 meters in scale. So you have to remove the sedum, but we're pretty good at removing things we don't want. And so that allows us to do lake ecology. So, yeah, I know. It's like, what do you want to do? Come to the toy store. Oh dear, my husband.
Great talk, sweetie, great talk. The questioner, a couple of questions ago, talked about your influence, the work that you're doing, how it influences other fields. Mm-hmm. And you mentioned toxicology, mm-hmm. Dinophysis, and Alexandria, and other species. Mm-hmm. Do you want to say something about, well, I mean, about global warming and what your work means to the shellfish industry, for example, and people that rely on I
Yeah.
mean, this is the big thing, and
It is.
it impacts big fisheries. So,
Yeah.
you know,
So being able to identify phytoplankton that are toxic or harmful is, of course, really critical. And sometimes we can see them because they're very distinct and they come in very high concentrations. Other ones, like say Alexandrium, which we get around here, shows up with all the other dinoflagellates, and it doesn't show up in very high concentrations, but it's exceedingly toxic and causes paralytic shellfish poisoning, which has huge implications for the shellfish industry. And so part of the sort of validation part of the project that I talked about was looking at microscopy and being able to monitor phytoplankton species with a microscope, essentially that's measuring all of the time, and developing algorithms to identify the appearance of these toxic species, figure out what the conditions are, why they show up, when they show up, and who are they showing up with. And so that provides us with the capability of being able to give early warning to aquaculture systems, and also potentially being able to predict based upon the conditions that allow them to grow when they might show up. And so I think that's your question. Yeah, thanks. All right, Mark, last one.
Well, it's kind of fun because several of my earlier questions were answered or addressed already, which was nice. I was going to ask about the thermal structure in the ocean and the implications for it, but you answered that to a large degree with Allison's question. But I'm wondering if you can use this tremendous palette of 200 crayons to look up the food chain at other organisms, zooplankton or other things, and see that trophic structure changing as they populate the ocean and change its color, coccoliths or whatever else, zooplankton.
So I only talked about absorption, and I didn't really talk about backscattering. But backscattering is just the redirection of light. Light comes down, gets scattered in the backward direction. It turns out that it's really dependent on not only the composition, but the size of the particles. And so we can actually do a model that inverts to figure out the size of the particles that are suspended in the water. And once you're away from sediments, like away from the coast, those particles are phytoplankton, zooplankton, anything in this trophic system. And so we can look at changes in the size structure of these organisms. And so you can begin to see whether we're looking at like big fat phytoplankton eaten by big fat zooplankton eaten by fish. And that's a very short trophic structure. It's 3 steps. Versus when you have very small phytoplankton, which are eaten by small zooplankton, and maybe eaten by a little bit bigger zooplankton, and then small fish, and then medium fish, and then finally you get to like a whale. That's a lot of steps in that trophic system, and that's a lot of sort of lost energy and mass and carbon. And so the ecosystems that are short versus ecosystems that are long have tremendous importance to think about in terms of fisheries, in terms of carbon, et cetera, and we can do that too. All right, thank you very much.